Hi, my name is Lola Henninger and I'm a part of Dr. Sterling Sudwig's Cellular Biology Lab. Alongside my colleagues, Matthew Anderson and Sarah Alvarado, we would love to do a presentation on our research technique, the 2-electrode voltage clamp electrophysiology recording. The voltage clamp technique is a powerful experimental method used to study ion channels. It works by injecting current into a cell to keep the membrane potential fixed at a chosen voltage, allowing us to observe the cell's activity at that voltage. This lets researchers measure the flow of ions through channels in response to physical, chemical, or ligand-based stimuli. Voltage clamp recordings have been essential in neuroscience, famously used by Hodgkin and Huxley to uncover the ionic basis of the action potential. Today, they remain a cornerstone of pharmacology and electrophysiology, helping scientists study how drugs, infections, or receptor modifications alter ion channel behavior. This information helps identify potential molecules that could modulate flow through channels. In our lab, we use the voltage clamp technique to measure current through nicotinic acetylcholine receptors, which express throughout the human body. The subunits we are specifically studying are commonly co-expressed in the hippocampus, stimulating learning and memory, which can affect disorders such as schizophrenia and Alzheimer's. In our research, we are using the 2-electrode voltage clamp technique to find how we can stimulate those nicotinic acetylcholine receptors in the hippocampus to a degree in which we can therapeutically treat, through medication, treat Alzheimer's and schizophrenia and other such disorders affected through the hippocampus. We do so by injecting frog oocytes with messenger RNA encoding alpha-3 and beta-2 subunits of nicotinic acetylcholine receptors at different ratios, typically 1:5 or 5:1. Then 5 to 10 days after injection, we perform a 2-electrode voltage clamp recording at the holding potential of -60 millivolts. And each recording, we do a 1 millimolar acetylcholine application to elicit a response, then wash with extracellular fluid for about a minute and a half before doing 2 more recordings. And then using a variety of concentrations, we're creating a dose-response curve to eventually apply positive allosteric modulators that can increase the flow of these channels and increase our dose-response curve to hopefully, through medication, treat things like schizophrenia or Alzheimer's.